Okay, I think I can just move fast. So when you talk of DNA, this is double helix. Like I have stated, it is made up of two strands. So this side you can see RNA, and then this side you can see the DNA molecule. And then this DNA molecule, you need to understand that the reason why it has two strands is because of the fact that this thing is normally having hydrogen bonds. So there are hydrogen bonds between the nitrogenous bases. There are hydrogen bonds between the nitrogenous bases. And these two strands, they normally twist. They normally twist around each other, leading to the formation of a double helix structure. So normally DNA is double helix. Okay. All right. Let me quickly move on. So in this case, I'm just trying to explain the same thing, but using a different picture. So when you talk of the DNA, the nucleotide, when you talk of the nucleotide, you need to understand that there is a pentose sugar, okay? And this pentose sugar, if it is in DNA, then it is deoxyribose. If it is an RNA, then it is ribose. Then when you talk of the nitrogenous base, it can either be a purine or a pyrimidine. It can either be a purine or pyrimidine. So that's the nitrogenous base. Then the phosphate group, it's just the same way. There is nothing that changes there. All right. So what forms the backbone of this nucleic acid? That's the question we need to ask ourselves. You can see this nucleic acid being like that. So there is the components that forms the backbone of nucleic acid. So the bond between the pentose sugar and the phosphate group, they make the backbone of what? Of the nucleic acid. Like you can see there in blue, that's a nucleotide, and that nucleotide with the next nucleotide, they are forming a bond. And this bond is existing between the what? The pentose sugar and the phosphate group. If you check up the diagram there, there is the blue which is showing the nucleotide, and another blue which is showing the nucleotide. When these two come together, they are going to share that bond, and that bond is the one which is between the phosphate group and the what? And the ribose sugar. These two are forming the what? The backbone. Same applies with the next one. You can see the two nitrogenous bases together forming a bond there, and that bond, which is an ester bond, such is helping in terms of making the backbone. This backbone is very important as it helps us to understand the pH of DNA. Okay, it helps us to understand the pH of DNA, and it also helps us the behavior of nucleotide when they are in a neutral solution or when the pH is increased or decreased, which are very important for your exam. Yes. What's the question?
You are breaking. Repeat on
the backbone. Okay. The backbone of this nucleic acid is formed by the bond between the sugar and the what? And the phosphate group. For example, when you look at the blue guys, so the blue guys are having a nucleotide, and another blue guy is having the nucleotide. There is a bond in between, which is shown by the red guy there. That bond, it is the bond which is creating between the phosphate group and the what? And the ribose sugar. And that bond, it is forming part of the what? Of the backbone of nucleic acid. I hope that is clear. Is it clear? Is it clear? You're welcome. Okay.
Yes.
All right, thank you. So when you understand these things, I think I've already explained that, whereby the backbones, the nucleotide, are being added on each nucleoside. So the question is, why is this important? So make sure that you understand why is this important. Okay, so all this will be explained later on. Now, when you look at DNA molecule, I think I'm just repeating myself because I've already explained most of these things. So when you look at a DNA molecule, it is double-stranded. So since it's double-stranded, that simply means that there is a bond in between the bases. For example, there is a bond between cytosine and what? And the guanine. And there is a bond between adenine and the thymine. So you also need to understand, for the sake of understanding, you need to understand that at least 10 nucleotides make up a DNA strand. At least 10 nucleotides are very useful to make up a what? A DNA molecule. So one nucleotide, another nucleotide, another nucleotide, until they reach 10. That's when they can be said to be what? To be a DNA molecule. And there is a nitrogenous, a hydrogen bond between the what? The nitrogenous bases. This is because of the fact that there is NH bond, which is a polar bond between nitrogen and hydrogen in the what? In the purine and pyrimidine. And then when you talk of these bonds, they're able to create that hydrogen bond because these bonds are polar bonds. And when you talk of polar bond, these are just as a result of what we are calling the difference in electronegativity, such that some compounds such as nitrogen, oxygen, phosphorus, including sulfur, they tend to be selfish and they will pull the what? The electron towards their what? Nucleus, as compared to the vulnerable oxygen. This makes these guys to be partially what? Charged. So because of such, these guys are able to make the hydrogen bonds. Remember, there are 2 hydrogen bonds between A and T, and there are 3 hydrogen bonds between cytosine and guanine. So make sure that you take note of that. Is there any question before I move on? Any question? Any question? They will ask questions. Anyway, the next thing is, when you talk of this nucleic acid, like I previously said, these nucleic acids are just playing a role in the storage of what? Information. And this storage of information, it helps to be transmitted from the parent cell to the what? To the daughter cells. So take note of that. And this can just be related when you talk of what? A book. Just like a book. So a book is acting as a storage of whatever notes you are writing. I know some of you are not writing the notes, but when time comes when you'll be writing the notes, you're going to understand that you're going to use that book when the exam is about to reach. So that book is just acting as a storage of what? Information. So the storage of information in terms of human being, in terms of genetic material, that is the job of what? Of DNA, which is just a nucleic acid. All right. Now, This information is stored in what we are calling the genes. The genes act as a storage of this information. The genes act as a storage of this information, and this information has to be transferred from the parent to the what? To the daughter cells. Please switch off your mic a bit. Mumvela na kuli pompi. Anyway, next. Okay, so another nucleic acid we can talk about is ATP. So ATP, when you talk of ATP, ATP is playing a major role in the provision of what? Of energy. And this ATP can exist in so many forms depending on how many phosphate group it is having. Okay, it's depending on how many phosphate it is having. You can have triphosphate, you can have diphosphate, you can have monophosphate. Okay, so those are just part of the what? The way in which energy can be stored. This will be important when we talk about the biological importance of the study of nucleotides. Okay, so like I said, when you talk of ATP, whereby you have a nitrogenous base, a ribose sugar, and you also have 3 molecules of what? Of phosphate. So that's what you can talk about when you say the ATP. All right, so you can see there you have adenosine, and this adenine will react with a ribose sugar, and after that it's going to react with a phosphate group. In that case it will be what? Adenine monophosphate. Then when there are 2, it will be adenine diphosphate, and then when there are 3, it will be known as what? Adenine triphosphate, which is ATP. Okay, so make sure that all that information is something that you are able to remember. Okay, so I'll be a bit going back and coming back because of the slowness. So like I was saying, here you need to understand that the adenine will react with a ribose sugar, and that site it will be a nucleoside. After that they will react with a phosphate group, that's adenine monophosphate. After another group it will be adenine diphosphate, then after another group it will be adenine triphosphate, which is the ATP. And the main job for ATP is just to help in giving out energy. So let's quickly look at some few exam questions, though don't expect the exam to be this simple. This is first year. So anyone who can answer number 1? Anyone who can answer number 1? You can just unmute yourself. Huh?
The answer
Is C? Others are saying B, others are saying C. B?
B.
Okay. All right. The answer is just B. All right. That's correct. Okay.
I have a second.
Yes.
Oh yeah, so you were talking about ATP. So I wanted to know, like, for example, on the backbone. So like, is there like what can I say? Like a chance where like three phosphate groups. Like yeah, like that.
Okay.
Or just one, always one.
When you talk of ATP, is it ATP or DNA? ATP or DNA? Which one are you talking about?
You were explaining on ATP how it.
Yeah.
So I was asking, can it be in the backbone, or it's only one phosphate group that makes the backbone? Together with the sugar.
No, when you talk of ATP, the information about DNA and the information about ATP, they're a little bit different. When you talk of DNA, this is whereby a nucleotide and another nucleotide unite to form what? To form DNA. But when you talk of ATP, okay, this is whereby a nucleotide, like this nucleotide, instead of it adding another nucleotide, it's going to add what? A phosphate. Instead of it adding another nucleotide, in this case, it's going to add a what? A phosphate. So in this case, let's say the formation of what happens with the formation of ATP. When this nitrogenous base react with a pentyl sugar, react with a ribose sugar, they are going to form that bond, and then this guy will react with a phosphate group to form what? AMP, which is the adenine monophosphate. Then instead of this reacting with a sugar to form a DNA or RNA, in this case, this guy will react with a phosphate group to give the adenine diphosphate. So it's not forming any backbone. It is just the addition of the phosphate group. And then there, it can also do the same thing. Is that clear? The lady was? Okay. Let me quickly go on the next part, which was the question part. That one has been answered. Let's quickly look at that. What is one difference between DNA and RNA? Anyone? Okay. I can see B busy.
B.
Okay. We'll go for that, which is correct. All right. Let's go on the next one. What are the three parts of a nucleotide? What are the three parts of a nucleotide?
C.
Others C. Others?
C.
Right. C. Correct. Okay. Now, this is how you shine. This is what you can shine. But this is not. B is not correct. 3 carbon sugar. Meaning there are 3 carbons. There are 3 chain. 3 chain sugar. Meaning it is not a furanose. Okay? That's why that 5 is there, not 3. Okay, so guys, listen. This is what your mind is telling you to know. But this is too easy for anyone. This is too easy for anyone at your level. If you find such a question, praise be to God. But the fact is that these are too easy for anyone. These are some of the questions that you need to expect in your exam. A question like that one. Foot and mouth disease virus is a pathogen that causes foot and mouth disease in livestock. An analysis of the genetic material of the O strain of FMDV showed that it contains 1906 adenine bases, 2131 guanine bases, 1642 uracil bases, and 2365 cytosine bases. Which one of the following describe the genetic material of the virus? So such is what you need to expect. What's your answer?
B.
Others D, others B. B. So the answer is B. Why? Because there is an issue of uracil bases, and you know that RNA is single. Congratulations to all those who have shined. May God bless you. But let's go to the next one. This one is too easy. Which of the following supplies cellular energy for muscle contraction and nerve impulse? What's the answer? C. All right, people are shining. C. Let's go to that one. Let's shine together. Gemza, an analog of deoxytidine, promote apoptosis during pancreatic cancer therapy. Which of the following is true about this nucleoside analog gemcitabine? What's your answer there now? Let me even go to the messages. Others D. I can't see the messages. People are not sending messages. What's your comment? What's your answer there? Just one.
No, it's that's not one. There are more answers.
Yes, there are more, but give me the correct ones. C. Okay. Which other one? Okay.
And C might be correct.
G looks wrong. Why is A correct? Why is B correct? Why is C correct?
I think A is correct because when you take it by mouth, it goes to the liver where it's phosphoric. So it's correct.
Okay. B? Why is B correct?
Nucleoside analog.
Who has raised the hand? Brivia. You can just unmute yourself and so I'll leave this question the way it is. So this question, it is one of the application of what? Of the study of nucleotides. This is just one of the application of the study of nucleotide. So when you talk of this question, it is one of the common questions you are going to meet, and it's a question which you need to understand and analyze. So let's quickly look at another one. So make sure that you take note of this question. In the next class, when I'm done when I'm done the topic, I'll get back to this question and I'll be able to explain it. I'm going to get a lot of questions of this type, not these ones, Simonsa, not these ones which you have swatted. This is likely not to come. You might not even meet it anywhere. This is how biochemistry comes. Which of the following is a nucleoside analog that would be used in treatment of HIV using HAART therapy? Which one can you go for?
Yes.
A and B? A and C? Muriasanga mansa ya bufimu bachai. Anyway, we'll talk more. We'll talk more about this when we are done with the topic. Take note of this question. I have about 6 of them of this type. So after we are done, we are going to look at such questions. So these are questions you expect in your test, not O2. This one, it's most likely. This, forget. Such, forget, anyone can get it wrong, correct. These, anyone can get it correct. What you need to expect much are questions like that, questions like that, questions like this one here. These are questions that have to be at your fingerprint. Anyway, we'll talk about these questions when we are done with the topic. So we talked about the what? The components of the nucleotides. So one component we can talk about is the nitrogenous bases. So when you talk about the nitrogenous bases, this can either be purine or pyrimidine. So you need to know this can be either purine or pyrimidine. So when you talk about pyrimidine, these pyrimidine are 6-membered heterocyclic aromatic rings containing 2 nitrogen atoms. And then when you talk of purine, purine are a combination of a pyrimidine ring with a 5-membered imidazo ring. What do you understand by imidazo ring? Anyone? What do you understand by imidazo ring? I only have 12 minutes to be done. What do you understand by imidazo ring? And what do you understand by heterocyclic aromatic ring? Hey, people, unmute yourself. Yes, Mufuka. Guys, you should be jobbing in detail. Let's be jobbing in detail. Please, let's be jobbing in detail. It's not just about knowing on surface, whereby you just know, oh, the nitrogenous bases can be purine or pyrimidine. No. Please, let's be jobbing in detail, whereby you know every content. When you talk of biochemistry, I can assure you, it's all about knowing every point, especially your lecture notes. Every point. You don't even need to stretch much when it comes with biochemistry. Your lecture notes are very important. So, those lecture notes you have, when you have them, the next thing your job is to research each and every point in those lecture notes. Then you are done. Anyway, let me move on. Then when you talk of purine and pyrimidine, you need to understand that these guys are relatively insoluble in water. I'll pose a question after that. These guys are relatively insoluble in water, meaning they are hydrophobic. Now, my question is, what happens when the pH is increased? What happens to the solubility of the purines? Does it increase, decrease, or it remains constant? Anyone? We are saying the both purines and pyrimidines, which are nitrogenous bases, are relatively insoluble in what? In water. That means that they are hydrophobic in nature. The question is, when the pH is increased, what happens to their solubility? Does it increase, decrease, or remain constant? Are you guys getting me? Anyone to confirm?
Yes, sir, we can hear you.
Yes, what's the answer now?
Since the OH groups are mostly replaced with other elements.
Okay. All right.
When the pH is increased, those
Okay, so when the pH is increased, someone has said that the solubility will increase. What about when the pH is decreased? When the pH is decreased, will the solubility increase, decrease, or remain constant? When the pH is decreased, will the solubility of these nitrogenous bases increase, decrease, or remain constant? Eh?
it will remain constant.
Okay. All right. Okay. So, when the pH is neutral, the nitrogenous bases are partially insoluble in water. In short, they are hydrophobic. Now, when the pH is increased or decreased, these guys, they tend to be soluble. Why? The answer is simple, because they normally become charged. Because they normally become charged. That's a reason why in a pH which has been increased or decreased, their solubility increases. I hope that has been understood. Okay, so let me now explain on the two terms that I asked you guys. The first term I asked you was what? Heterocyclic compound. So when you talk of an heterocyclic compound, it's just an organic compound in which one or more of the carbon atom in the backbone of the molecule has been replaced by another atom which is not what? Carbon. For example, when you talk of a purine or pyrimidine, one part has what? Two parts actually. They have what? A nitrogen atom. So the other term which I asked was, what does imidazole mean? So when you talk of imidazole, this is just a planar 5-member ring that exists in two equivalent tautomeric form, because hydrogen can be bonded to one or another nitrogenous atom. What do you understand by tautomeric atom forms? Like when you talk of 2 tautomeric forms of adenine, what do you understand by that? What do you understand by that? The 2 tautomeric forms?
Isomers maybe.
Yeah, they are isomers, correct. What kind of isomers? What kind of isomers? So I only have 4 minutes. Okay, let's move on. I'm going to explain the type of isomers as I move down. So, when you talk of purine, purines are a natural substance found in some food. But this purine, when it is being reversed, it is capable of producing what? Uric acid. And this uric acid, when it is kept, and this uric acid, when it's built up, it's able to form uric acid what? Crystals. And these crystals, they are normally not good for your health. As a result, this can lead to what? Certain healthy issues. So when we start looking at the questions in the next meeting, I'm going to talk about some questions which will need this as a reference. So that's the structure of a purine. You need to understand that a purine has how many? Has 2 ring structures. These numbers are not for decoration. These numbers are not for decorations. They are important. So examples of purine you can talk about is adenine and the guanine. Adenine and guanine are examples of purines. Okay, pyrimidine, you need to understand that pyrimidine, it is just a single ring structure. And when you talk about pyrimidine, this is just one of the nitrogenous bases which forms part of the DNA and RNA. Examples of pyrimidine, like you have cytosine, which I've shown there in terms of picture. I've also thymine, which I've also shown in terms of the picture. There is also uracil, which I've shown in the words in the picture. You need to understand that DNA has thymine and not uracil. And then RNA has uracil and what? And not thymine. Meaning, one of the differences between the two is caused by the type of pyrimidine they have. One of the differences between DNA and RNA is caused by the type of pyrimidine they have. Alright, so please make sure that you understand all the content. Let's quickly look at properties of nitrogenous bases. One problem I've found with most of us is that we overlook certain causes. But trust me, every cause is having one person who is repeating that cause. That should tell you something. Please don't underestimate or overlook any course. It might cost you. And you are in a competition of over 2,000 students competing with one sport. 2,000 students competing with one sport. Amongst the 2,000 plus, there are those who are taking only one course, and there is you who is taking 8 courses. And those who are taking at least 1 course or 2 courses, there are people who have done it before, and there are people who can easily understand. And I'm sure you can't tell me that you can compete in terms of balancing courses. You are in a very tough competition, and there are people who can just make it. Okay, let me not talk much. Let's look at properties of nitrogenous bases. We need to understand that every nitrogenous base shares one feature, and this feature is that they have a six-sided ring with 4 carbon atoms and 2 nitrogen atoms. 4 carbon atoms and 2 nitrogen atoms. However, you need to understand that a purine has an addition of 5-sided ring created by 1 more carbon and 2 more nitrogen atoms. Then a pyrimidine has only a nitrogenous base, has only 1 6-sided ring. Then when you talk of the properties, they are strong, ultraviolet, light absorbent due to the aromaticity of their heterocyclic ring structure. Because of the presence of that nitrogen there, these guys, they tend to absorb what? UV light. Remember what we said? When you talk of heterocyclic ring, these guys, they have what? They have replaced they have replaced at least a carbon in their ring. So these nitrogenous bases, because of that properties, these guys tend to absorb UV. They are weakly basic compounds. Then these guys are hydrophobic and relatively insoluble in water at the near neutral pH of the cell. That means that when the pH is increased or decreased, these guys, they are soluble because they tend to be charged. Then there is hydrogen bond between purine and pyrimidine bases. And this is very important in the formation of the double helix structure of DNA. Remember, DNA is double helix. This is due to the hydrogen bond which is shared between a purine and a pyrimidine. Take note of this part. We are going to talk about it in terms of questions later on. And I'm emphasizing on one thing. These questions, they are a little bit in detailed. Okay. Then they are able to undergo keto-eno-tautomeric shift. So when you talk of these guys, they are able to shift. They can be a keto or a eno type of isomer, and this is because of the electron having an OH and NH2. This will be explained in terms of a picture on the next slide. So keto-eno-tautomeric shift, for example, if you look at thymine here, this is the area of interest. So this keto is changing into an eno form. These guys are what are said to be tautomeric form. They are existing in tautomeric form. And then you have this guy in terms of guanine, which can change into this one to that one. Okay, there is an important point I've raised here, which is just what I have explained. So these two are just the tautomers of each other. Tautomers of each other. That simply means that they are isomers. By the type of isomer, it is what? Tautomer. Okay. I'll give an exam question about this, and people should be able to answer it. Okay. The next one we can talk about is the pentyl sugar. So when you talk of the pentyl sugar, this is one of the part which brings the differences between RNA and DNA. When you talk of RNA, it has an hydroxyl group on carbon number 3. The number of carbon should be noted. So when you talk of 2 carbon, this 2 is because we just want to differentiate the carbon in the ribose and the carbon which is in the what? In the nitrogenous base. That's why there is a cap there. But it just has to be 2 carbon. But because of another carbon in the nitrogenous base, we normally put a cap. So on 2 carbon of a ribose, there is an hydroxyl group, and on the 2 cap of DNA, there is what? There is no hydroxyl group. That picture can easily show RNA ribose. So this is the ribose sugar. This is the ribose sugar because at this group there is OH. This is the deoxyribose sugar. As you can see, at this group there is no OH. Okay, what else can we look at? So when you talk of the pentose sugar, this is a 5 carbon sugar. I'm sure someone had to get confused at one point at one of the questions. I think that has been explained, and that has also been explained, and this has also been explained. Okay, one thing that I haven't explained is that both pentose sugar present in the furanose 5-membered ring. That's why that multiple choice are the 5C. So 5-membered ring. A 5-member ring is known as furanose. But you need to know how many carbons are in a furanose ring. Okay. Phosphate group, which is like the cross for Christians. So this is a phosphate group. So when you talk of a phosphate group, what things are you supposed to take note of? A phosphate group has 4 oxygen. The phosphate group has 4 oxygen. Only 1 oxygen is what? Is the double bond. The rest are single bonds. So these single bonded oxygens are the ones which are more reactive. The single bonded oxygens are the ones which are more reactive. I think that has also been explained. In a case whereby this has no double bond, whereby all of them are single bonds, then it's not a phosphate group, but it is a phosphate ion. So this is where the charging is coming from. Okay. All right. I think I am going to end here because of time, but I was supposed to look at all these things and also look at biological important, and I'm sure I had a lot of questions that were popping up. So there is a lot of information that I was supposed to share, but I'm going to give you an assignment of explain at least 4 functions of nucleotides. So please make sure that you can submit your answers in my inbox if you have time. And then for those who would want to get in touch, I'm sure my line is everywhere. You can simply WhatsApp me, you inbox me. It's not too late to join us, and it's not too early to join us again. The decision is up to you. So guys, for today, I think I'll end here unless if there are any questions. Otherwise, there is a lot that was supposed to be explained, but due to time, I can't manage. Any question?
How much?
It's 80 kwacha each course. 80 kwacha each course. That's the costs.
No discount.
If you want a discount, come to my inbox. I'll give you a nice discount.